Hi, I'm Jim Smith. I'm Deputy Director of the Division of Metabolism and Endocrinology Products in the Office of New Drugs at FDA. Quite a panel to follow. That's a tough act. Alex's bucketing of ensemble efficacy and utilization factors I thought was great and raises a lot of questions in my mind about where FDA role sits, and that could be a discussion later. As a disclaimer, my comments are my own only and certainly are not meant to represent the views of the agency. As I thought about blinding in the context of real-world evidence, I really kept coming back to the fact that what I think about blinding in this context is really no different than what I think about blinding considerations for traditional clinical trials. The most of the considerations are really the same. The goal in general is to reduce the likelihood that study participants and those involved in the management will not be influenced by the treatment assignment. And so I sort of reflected on, when is that particularly important? And several of these factors have been already mentioned. Subjective endpoints. When we have more objective endpoints, we're might be more comfortable, but I'll say a bit more about that in a second. If we expect a relatively modest effect size, and Dr. Califf mentioned earlier today that he thinks that is probably the majority of cases, especially for common diseases that afflict the large part of the public health, and I totally agree with that. But let me also give a caveat that we could take an objective endpoint, like let's say blood pressure, that is just an automated measurement by a machine, and we could pair it with a large treatment effect, like was seen in early renal denervation trials. And then we can become incredibly surprised when we have a sham-controlled trial where we see no difference between the intervention and sham control. So I don't think that we should immediately say, large treatment effect, objective endpoint, we're good. This could be an open label uncontrolled trial. I think that we still need to walk through the exercise of where could we go wrong. Biology always humbles us. We would be concerned about blinding when we think there's a reasonable chance that patients could be treated differentially based on treatment assignment in a manner that could influence the outcome. Take an objective endpoint. Once again, blood pressure or hemoglobin A1C is measured. There are open label trials conducted in this space, but one must always worry about, is the control arm really being treated as aggressively as the experimental arm? And that's not always easy to tease out, right? Anyone who's analyzed concomitant medications and doses, it's very harrowing, and you're never really confident that you actually know what happened to patients in a clinical trial. Endpoints that require a subjective decision by the provider prior to the event being referred to an outcomes assessor. So a lot of comments have been made about blinded outcomes assessors. They only assess what has been given to them. So let's take a hypothetical example where you have an outcome that is based on a blinded pathologist's read of some sort of a biopsy specimen. The pathologist has no idea what the clinical course is. They have no idea what the treatment decision is. But who made the decision to conduct the biopsy? What if it was not a protocol-driven biopsy, but rather a biopsy that was driven by a clinical suspicion that a lesion was worsening or a clinical suspicion that a patient was, say, rejecting an organ? That is now a decision that is made by somebody who, if they are unblinded, it does not matter that the outcomes assessor is blinded or not, because the bias has already been introduced even before the outcome got to the outcomes assessor. So outcomes assessor blinding in and of itself should not just be thought of as a panacea that we've taken care of bias of outcome assessment. Situations where knowledge of the treatment assignment could influence whether a patient adheres to therapy or a provider's recommendation to withdraw a subject from a trial. And that's been touched on a little bit already, and I won't elaborate on that. However, I do acknowledge that there are situations where the ability to blind is perhaps just not feasible. We have seen drugs where there's essentially 100% severe injection site reactions that only occur with the treated therapy, okay? Or another drug that I can think of where essentially severe GI intolerance was essentially seen by 100% of the patients, whereas that's not going to occur in the placebo arm. There's just not a way to deal with that. And it just has to be acknowledged as a limitation when you're reviewing the data. And then whoever is reviewing the data has to decide how much they think that could affect the outcome. And so we should remember that the bias associated with treatment assignment is often subconscious, right? And both the direction and that magnitude of bias is difficult to predict. So one challenge that I had as I looked at the decision aid is to predict, what this bias is going to be. Well, by its very nature, that is incredibly difficult to do. It's unmeasured. Patients could believe that this is a novel treatment that is really going to help them, and therefore they will overstate their benefit and they will understate their adverse event profile. Similar patients in the same trial might have providers that believe that the status quo is perfectly good, yet some company wants to try this new drug. Do you want to enroll in the trial? And they could come at it from a very different perspective. And I don't think we know that. That's not captured. So it's very difficult to predict how bias is going to creep in and what direction and magnitude it's going to be. So blinding is the mechanism to reduce the possibility of that bias, whether it's unintentional or intentional bias that would affect the trial's results. So when I looked at the framework and to go through the little buckets, what expectations or preferences providers or patients be expected to have. Like I said, I think that could be very difficult to predict, but it's a question worth asking. I just don't know how confident we can be in our responses to that. And that could very well be therapeutic area specific, and there could be some areas where it's far more well known than others. That could even be influenced by other investigational products that just temporally in time happen to be available. I mean, if a PI is involved in 3 clinical trials at a specific time and is really excited about one of them and not so excited about the other 2, but they feel they have to enroll patients in all 3, that they could get a result from those 3 trials that if that same investigator participated in those 3 trials spaced out in time, it could be very different. In the second bucket, for similar reasons, how would those preferences or expectations influence intervention uptake or adherence, or the intensity with which a treatment is delivered, or the likelihood that beneficial adverse events will be reported or observed? Once again, I think it depends, and we might not always know. In part, the fidelity or intensity with which an intervention is delivered, it's not only the intervention of the experimental agent, but it's also the co-interventions and the treatment of the control group that would be important to consider there. In the third box of how might those expectations or preference differ in the settings where trial results will eventually be applied, well, when the results are eventually applied, if the trial is believed and if it's well done, then that trial itself could affect the expectations and preferences of the future. So that's difficult to predict. And once again, it could be affected by available treatments in the future setting, which might even be a different health system. And that in part is a challenge in my mind about the ensemble efficacy and utilization factors that Alex put up. I generally think of FDA or the FDA's review of drug labels as we're trying to give what is best known about the effect that a drug is purported to have and its safety. And that's the ensemble efficacy that Alex described. Once you interject all the things that can happen in a health system to then generate the real world effectiveness piece, that is not going to be the same in every health system in the country. And we're trying to serve the country and the public health. Imagine this, anticoagulants are not my forte, are not my area, but it's not a stretch to think that if one conducted a trial involving warfarin, it's in centers where they had a very inefficient and poor mechanism to monitor INR. You could get a very different result than if you conducted that same trial in a center where they have very strict protocols and an efficient system for INR monitoring and managing their patients. And if you got a poor result in that first trial, would it really be fair to blast warfarin in a competitor's label? I don't know that would be the case. So those issues would have to be very, very carefully considered. How might concealing treatment allocation from patients and/or providers reduce biases due to preferences or expectations? One would hope that concealing treatment allocation would reduce the bias, as I've discussed. But I believe that it's going to be difficult to determine a priori what the magnitude and the direction of the bias is that you're actually getting rid of. It is possible that you could obscure meaningful differences by blinding a trial. And that, in my view, depends on the research question. For example, if one is interested in whether or not it's easier to get patients to adhere to an injectable therapy or to an oral daily tablet, issuing a double dummy so that both groups get the same treatment administrations is not going to help you answer that question. But that's a different question, right? So it really depends on the question that you're wanting to answer. And then the last box of how might procedures necessary to conceal treatment allocation from patients or providers impact the acceptability of trial participation to patients or providers. I suggest ask the patients. I won't comment on what area it was, but we were dealing with a rare disease where we were hearing consistently that it would be impossible to conduct a placebo-controlled trial in this population. We had a meeting with the patients outside of any sponsor, and uniformly the patients told us, if this will give you the best data and we could go on to a placebo for 6 months, we are happy to do so. So ask the patients, because we might always not make the right assumption. The cost of the trial could certainly increase by blinding, but I would argue that if an unblinded trial gives you results that nobody believes or wants to interpret, then you've just wasted a bunch of money. And the risk-benefit ratio, I thought Alex made a good point here, especially with regard to sham surgery. And I'm not very familiar on the device space, but I could see that this could crop up there substantially. But I do come back to the renal denervation example and say that we ought to be really sure that we can't do at least a controlled trial before we bail on it altogether. So I will end there. Thank you.